We're taking a look at viruses and prions today with our Unit 1, Lecture 7. Viruses are one of the most common types of infectious pathogens. So what we want to do in Part 1 is take a look at kind of what they are and what they look like. And then in Part 2, we'll focus more on how they cause disease. And then in Part 1, we're also going to talk about prions, which are kind of related to viruses. So getting into it here, viruses are an interesting type of infectious agent because they're more like molecules than they are actual other types of infectious cellular agents. And if you recall from chapter 1, we call viruses acellular microorganisms, along with prions. So they're non-living. That's the idea of acellular. They're more like an infectious particle. Now, part of that is arbitrary, and there's technical reasons why they're not considered living. Kind of goes outside the scope of this class to fully investigate what exactly is living and why don't viruses fit that description. But they're not fully made up of all the characteristics of life. So they don't have cells, they don't necessarily metabolize on their own. But what's interesting is that when they hijack a host cell, they can get the host cell to do these processes for them. So a host cell becomes a vessel for their needs. And so that's why they fit this interesting spectrum, or an interesting position on the biological spectrum, which show next here. And that the idea is that, generally speaking, life is made up of atoms, which form simple molecules, which then form macromolecules. Macromolecules, by the way, would be things like proteins and carbohydrates and fats and lipids. Your DNA and your RNA, these are macromolecules. So life, these are kind of like the building blocks of life. Life begins with cells, and then for more advanced organisms, progresses up in this fashion. So you kind of build and then organize into different forms of life. Where viruses come in is really right in between these two. So they are not cells, they don't have the complexity of a cell. They can't be classified as a cellular organism, but they are more complex than your proteins and macromolecules. Macromolecules don't typically behave like a virus. So they're in this weird position kind of between life and non-life. And there's a lot of philosophical debate about what to do with that. Should we reclassify life, or should we try to reestablish the different parameters? But that's not relevant necessarily for our discussion here, but it does bring up a lot of interesting questions. What exactly is life? Now, from an infectious standpoint, viruses are everywhere. They are capable of not only infecting you and I potentially, but actually any part of the living world. Any living thing on Earth can theoretically be infected. So they're very common. They're all around us. And they're able to theoretically infect all life forms. But luckily for us, and for most life, each virus is sort of relegated to its own kind of host. So a virus will usually infect kind of a specific type of organism. So there's viruses all around us, but they're all specific to different organisms, and only a fraction of those infect humans. So we're kind of in this environment with viruses all around us, but they have different targets, and they're not necessarily looking for us, not all of them. Now, from certain types of viruses, like animal viruses, they may be able to move from an animal to a human. And in that regard, there is some flexibility. We see viruses mutate, and they kind of go from one host to another, or they change the nature of infection. So there is a little bit of leeway from one type to the next. But most of the time, a virus is confined to a specific host and doesn't change dramatically. So for example, there are plant viruses, but those don't ever mutate or jump to humans. So they stay specifically within the plant world. They might infect a different kind of plant, but they're not going to modify from plants to humans or plants to animals or vice versa. So every life form has its own kind of specific type of virus, and there's a little bit of flexibility and modification within those categories, but they tend to stay within their group. So that, just to clarify, that means that there are plant viruses, fungi viruses, algae viruses, protozoa, bacterial viruses, and many more. So there are literally different types of viruses for each one of those categories. So that's kind of what I mean there. Now, viruses are extremely small. They have such small dimensions that they are a fraction of the size of a typical cell. We talked about cells early on in the semester and how small they are. So these are a fraction of the size of a cell. Now, there is a lot of variation from one viral size to the next. In other words, the biggest virus is quite a bit bigger than the smallest virus. But they tend to be in this spectrum here. This kind of just gives you some averages about common sizes. So if this is a human cell, this is a bacterial cell, these are about the range of typical human virus sizes. And this, by the way, here, this little green thing is a virus that infects bacteria. So viruses that infect bacteria are about that size. That's called a bacteriophage. So if a human cell were a basketball, a virus would be about the size of a pinhead. So with that being said, these represent typical human-sized cells in green. These represent bacterial cells. And this gives you a better spectrum of viral sizes. So this is a small bacteria here. So these are average bacteria, small bacteria. And this, the Mimi virus, is a big virus. That's like a really big virus. They've actually discovered one that's even bigger. But at the time this was published, that was actually the biggest one at the time. So it's much larger than average. And down here, you have a molecule. That's like a macromolecule. Hemoglobin, by the way, is a huge protein. So this is a big protein. Okay, so that kind of gives you the spectrum of size there. I don't make you memorize that for the test, but that just kind of gives you some visuals of what we're talking about. These are real microscope images of viruses. You don't usually get a ton of detail, and these are super high resolution microscope pictures. So just to get this level of detail requires a lot of sophisticated machinery. And that's about all the more you can see. They're just little sort of seemingly simple shaped structures. So when we get into what we're trying to see there, illustrations, give you more detail. So a virus starts off with the capsid, or we'll start off with the capsid. And as it says here, viruses are kind of minimalist. They only need parts to invade and control. A capsid is almost like the body of a virus, if you will. And it's this core kind of light blue structure here. What's packed inside that, because this is meant to show that it's hollow inside, or at least there's an internal cavity there. And then what you'd find inside there, the central core, would be primarily the nucleic acids. These would be like your DNA and your RNA. And then some viruses will have additional proteins and molecules tucked inside there as well. So you kind of have the core, the outer shell rather, the capsid, and then the central core. And then another key part of a virus are what are called spikes. Now these are shown more dramatically in the illustration to make the point, but they're these protrusions that come out of the capsid in this case. And those, as we'll see, are key for attachment. In order for the virus to grab a hold, you absolutely need this key little feature here called the spike. Now, if we're looking at a layering system, or a layering diagram rather, a viral particle has its outer and inner core, and then these are the layers to each of those cores. This is kind of a reference figure from the book you can look back up. So on the exterior of a virus, you have the capsid, and you can kind of think of the capsid as like the body of the virus, and it's there to protect the central core, which would be your nucleic acids. Capsids are made up of individual units called capsimers. These are like the little pieces that put together the whole, which is the capsid, excuse me. Capsimers are the building blocks to the capsid. And depending on how those capsimers come together, you'll end up with one of three general types of shapes: helical, icosahedral, or complex. helical comes out as kind of a long cylindrical shape. Icosahedral will kind of look round in a lot of figures, but it's actually fairly specific. It's got 20 sides and 12 evenly spaced corners. That's how you get an icosahedral shape. And then complex doesn't quite fit into either one of these two. I call it the junk drawer of viral capsids because there's a lot of variation amongst those. So if they don't fit into one of these two here, they kind of go into the complex category. So that one's not quite as consistent. But when you look at these, you'll see illustration and then microscope image. Illustration, illustration, microscope image. And these are just some ideas of what we're looking at here. So these down below, these are the real images here, and then these are kind of how you put together an illustration. So each one of these, you can see it better in this particular one here on the helical. Each one of those little circles would be a capsimer, and then the way those all lock together creates the structure of the capsid. A little harder to see it on these other ones here, but if you notice, they all are kind of diagrammed to show that there's like individual components there, which are going to be the capsimers. Here, these are actually labeled on here. So anyway, that's kind of the way a virus comes together. It's pretty minimal. So as far as the body, so to speak, the capsid, you got these building blocks, and they sort of form these various patterns. Now, what goes inside of it are the nucleic acids, which are the key kind of information-bearing structures of the virus. When you compare, when you put those two together, capsid plus central core nucleic acids, they call that unit of the two together the nucleocapsid. Now, pretty much all of your major viruses have at least this level of complexity. And then a lot of animal viruses, not all of them, but many of them, will actually have another layer of complexity, which is a little hard to see in this diagram, but this is called the envelope, and this is a characteristic of enveloped viruses. And as it says here, most human and animal viruses have an envelope, or in other words, they are envelope viruses. So this over here is showing a naked virus, and you can kind of see, which a naked virus, by the way, has just the capsid and the nucleic acids. This structure here. So you can kind of see this icosahedral capsid shape here. And then over here you can see kind of a similar shape. That's the capsid, and then an envelope covers all of that around the exterior, and then the spikes stick out of the envelope like that. So an envelope virus kind of takes all the core features of a naked virus and incorporates those with an extra layer called the envelope. And that's how you get an envelope virus. And that's like how the influenza flu virus comes. That's how your COVID virus comes. So a lot of your seasonal type viruses, flu, COVID type viruses, have this type of structure with the envelope around the capsid. Now, so far we've talked about the capsid and then the envelope there. Spikes, which will stick out towards the exterior. Let me just go back real quick. They'll always be there. If it's a naked non-envelope virus, then they'll stick out of the capsid. If it's an envelope virus, then they'll stick out of the envelope. But either way, you'll get spikes. And spikes are essentially attachment vessels for the virus. And the way a virus works is it uses those spikes to stick to the host cell. And it does that in a way that makes it fairly specific. So on the exterior of a host cell, you will have little receptors called glycoproteins. And what the virus wants to do is get that spike and to stick it into a host cell glycoprotein. And I'll talk about this in more detail in the next video. But the idea is that these are used for attachment, and they kind of stick out towards the exterior of the virus, and they bind to the host cell glycoproteins. And that's part of the key part of the infection process. So here you see the flu virus. This is influenza. And over here you can see a microscope image. This is a real image here of a whole bunch of flu virus particles right next to each other. And then over here we see an illustration of that. And what we're looking at is, it's shown to kind of peel back the layers. So these green studded structures, those would be the spikes. And then you've got the envelope, which is making up this structure around the exterior here, kind of shown in purple. That's actually the envelope. So the spikes are embedded down in the envelope. And then the capsid here, in fact, multiple capsids, are actually encased inside the envelope like this here. And so this is showing that you could theoretically have multiple capsids tucked inside of the envelope. And then the RNA in this case would be your nucleic acids. That would be like the central core. Those are now tucked down inside the capsid like that. So you've got kind of multiple layers, using the flu virus as an example here. Spikes, envelope, capsid, nucleic acids in the central core. So nucleic acids are a key part of life in general, and this is one of the areas where viruses and living organisms do actually overlap, and where there's an argument to be made that maybe a virus is a living thing. That argument comes from the fact that they use the same genetic letters as all of living organisms, which are your DNA letters and your RNA letters. And if you're not familiar with that, that comes up in the genetics section, which comes a little bit later. But in other words, they use the same genetic language. It's not a different language; it's the same one. So that's actually an argument in favor of viruses being considered life forms. But what's more relevant is that they'll use that to infect the cell. So viruses have the same genetic language, but it's a very minimal amount of information. And so they carry just a few viral genes in many cases that they're attempting to kind of get into the host cell, which, you know, when it comes to human viruses would be like you and I. They want to get the viral nucleic acids into a host cell. So when they do that, they have these genes that they can shuttle into the cell, get those kind of past your immune system. And if they can get that information into the cell, it can actually redirect cellular activity. And ultimately, the cellular machinery, quote unquote, can be hijacked by the virus to facilitate the needs of the virus, such as viral protein manufacturing and viral replication. So viruses carry the same type of information as life, don't have all of the processing machinery for it, so their goal is to get into your cells and use your machinery to replicate their own viral code. It's a lot like a flash drive or even a computer virus, where the information can be stored on an apparatus that requires a computer to run it. So when you talk about cellular machinery, one of the big examples are ribosomes. And this is something that viruses don't have. And if you go back to some of the key features of life, we talked about all life being made up of cells. And then we talked about the 4 minimal features of cells. And one of the 4 minimal features of cells are ribosomes. So in order to be a living thing, in order to function as a living thing, you need to have ribosomes. And that's part of your quote-unquote cellular machinery. So what's interesting about viruses, going back to their position on the biological spectrum, is they don't have ribosomes. So we can't really say they're just a traditional life form. But they know how to use ribosomes. So you have ribosomes in all of your cells, and the virus doesn't have its own. But if it gets inside of your cells, it knows how to use your ribosomes. And that's what makes them interesting. So that's an example of cellular machinery that they're going to take over if they infect your cell. So they kind of hijack the host cell. And it's a lot like a virus. A computer virus is code that needs an operating system to run. So a lot of the hackers are trying to get their virus onto your computer. That's why they want you to download something. So the information doesn't do any good until they get it into your computer. And then that information can take over and redirect the activities of your computer. So it's a lot like an actual real virus. In fact, a computer virus is named after real viruses. They're very similar in the way they work that way. So ultimately, the nucleic acids are the information needed to get into a machine operating system, which is your cell. And if you can get it in there, then the information kind of knows how to take over and redirect that cell towards its own ends. So we'll talk more about that in part 2 and how that causes damage. So it's very much like a computer virus. Your cell is the computer. The virus comes in and can redirect the activities of the computer to make it operate differently. And from there, you'll get disease. Kind of like you get computer problems, the diseases are sort of downstream, which we'll talk about in part 2, what happens there. Here, we're going to finish up on viruses and talk in the last part here about prions. But the last thing, a couple things about viruses is just a little bit about classification. Viruses can be either DNA or RNA. Here I'm going to talk about DNA viruses. DNA viruses can come in one strand or two. Some examples would be herpes, type 1 and 2, papillomaviruses like HPV, human papillomavirus, and adenoviruses, one that causes pink eye, respiratory infections, and things of that nature. So these are relatively common, and these are all examples of DNA viruses. Single-stranded and double-stranded are sort of variations of DNA. Double-stranded is more normal, typical of DNA as it occurs in a living organism. So double-stranded viruses their DNA is pretty much just like a living organism, whereas single-stranded is not like living things. So they have DNA, but it's not in the same structure. So they're a little bit different. So you have classification of DNA, single-stranded or double-stranded, and that's kind of where this figure over here shows you kind of how that breaks down. And it goes a little bit further, but those are some of the major broad classification levels there. So you also have then RNA viruses, so DNA viruses, and then RNA viruses. And the difference is whether they are infectious in the DNA or the RNA form. So these are both relatively common. Both types are relatively common. So RNA viruses can also be single-stranded or double-stranded. In living things, RNA is actually normal in the single-stranded form. So normally RNA is single-stranded, but there are some viruses that are double-stranded RNA. And then as far as RNA viruses go, there's another classification, which is plus sense or negative sense, positive or negative sense. And it's really just a sort of classification based on how many steps it takes for them to infect or take over the host cell. It goes a little bit outside the scope of this class, a little more of a nuanced detail, but it is a way of classifying. So RNA viruses will be classified based on single or double-strandedness and then positive or negative sense. So you could have, for example, a single-stranded positive sense RNA virus, for example. So common examples here, rhinovirus, common cold, influenza virus, that's your flu, coronavirus, that's COVID. And so these are pretty common, but those are some examples of the specific types that you would see here. Well, specific types are a little more specifically listed over here. These are different viral families, but these are some of the most common ones that we would see in humans. Now, another classification of RNA viruses are called retroviruses. Retroviruses are fairly specific. They are the viruses that have the ability to convert themselves from RNA into DNA. So they're RNA viruses, but they can actually convert their RNA code into DNA. And they actually do this inside the host cell that they infect. So this is like, for example, what HIV does. Lesser known, the human T lymphotropic virus, one that causes leukemia. But HIV being a little bit better known, one of the things it can do is infect the cell. So here you see it coming in. It's an envelope virus, by the way. So here's the spikes, here's the envelope, here's the capsid. That comes into the cell. The capsid breaks down and the RNA is released. So that's where we're at. We're at the RNA form. Now, in the HIV virus, what it'll do is take this little enzyme here called reverse transcriptase. So that comes packaged up inside the virus when it infects. That enzyme takes the RNA and converts it into a DNA form. And then in this particular virus, what it does is it takes that copied DNA and integrates it into your host chromosome, so your own cellular DNA, that is. And that process is called integration. It uses the integrase enzyme to do that. So what it does is it sort of comes in one form. Oops, sorry, excuse me. It comes in one form, copies to a different form, and then takes the copied form and actually embeds it into the cellular DNA. And this is actually one of the primary reasons why you can't get rid of HIV. And HIV becomes its own case study as to more detail about that. But ultimately, you can't really get rid of HIV. And one of the reasons why is because it integrates itself into your DNA, and it makes it almost impossible for the body to get rid of it. So anyway, those were retroviruses, and HIV is one of the most prevalent examples there. Last thing I want to talk about are prions, and prions are related to viruses in that they are both acellular microorganisms. So they both sort of fit into the non-living but still infectious and therefore relevant category. Okay, so prions are different than viruses, but they're similar in the sense, like I said, that they're not living but still infectious. And what prions are proteins that cause misfolding. And they do that specifically in the central nervous system of animals. And this is specific to animals, warm-blooded mammals, and that includes a variety of different types, but also humans. So a little more specific, we're talking about a specific kind of protein, and what happens is these prions cause misfolding of these proteins. And ultimately that misfolding ends up causing disease. And so it's fairly specific, a little hard to follow sometimes, but it's relevant enough that you just want to be familiar with it. This figure over here is showing a protein, and this is what a protein would look like in a illustrative form, not even necessarily what it would actually look like, but in illustrative form. So this would be a normal, what they call here the PRPC protein. So these would be proteins that are found along the central nervous system on nerve cells. So this is a nerve cell, and these are showing kind of where the PRPC proteins might be located. And this shows it in a simpler structure. This shows it in a little more of a detailed structure. What happens with the prion disease is a protein that's similar but different enters the body, and that is the prion. The prion is the different protein, and it comes in contact with these normal proteins through exposure, and it actually causes this shape to change and misfold or fold differently into this shape. So we go from this to this, and I know it looks like a jumbled sort of weird picture, but it's a different shape is the main idea there. And what makes that even more impactful is that when we get this shape here, normal to abnormal, not only does the normal function stop, the shape, if you refer back to what we talked about previously, shape determines function. Shape and function go hand in hand. So you change shape, you can change function. So not only do you change the function, which is not good, but this new folded shape now also becomes contagious and infectious to other types of proteins. And this is where I describe it here. It's like a domino effect. You're going to have these normal proteins scattered throughout these nerve tissues, and as one infectious prion comes in, it'll misfold one protein, and now that misfolded protein can go on to misfold more proteins. So every time a new protein misfolds, it becomes infectious to cause misfolding in other proteins. And that's the idea of the domino effect. So the disease is often slow in the beginning and progresses quickly towards the end. As more and more proteins begin misfolding, the effect becomes more and more pronounced because each new misfolded protein will go on to cause folding in other proteins. Now, ultimately, what you end up with is a disease process that manifests from non-functional proteins. So every time a protein misfolds, it stops functioning like it did in its normal position. And as you get more and more and more proteins that stop functioning, ultimately the disease progresses. And as I mentioned, this is part of the central nervous system. So it's key to recognize that the specific misfolding of proteins is occurring in nerve cells as part of the central nervous system. So as that system becomes degraded to protein misfolding, it simply stops working little bit by little bit by little bit. There's a lot more complexity to exactly what happens there. But for simplicity and not going into the detail, the central nervous system goes through a slow degeneration, and ultimately it manifests as almost something like an Alzheimer's or something like a dementia, although not exactly, but a sort of rapidly, more rapid, but kind of a gradual progression of a degeneration of nervous function. And usually by the time they've diagnosed it, it's getting towards the latter part of the disease. So what they'll usually do is diagnose it based on symptoms and then try to look for confirmation in the brain tissue, which will look like this. So the disease process is called spongiform encephalopathy. Over here on the left is normal brain tissue, and over here on the right is abnormal brain tissue. And you can actually see these white holes, and those would be some of the spongiform hole-like structures that these prions create. So the brain tissue is not exactly a one-for-one type of picture, but this is more or less what it looks like normally. This is what it would look like abnormally. And so all those little holes and structures are an area in the brain that's not going to be functioning properly. So as a prion disease manifests, that's kind of what's happening at the cell level. The brain tissue is dying. Now, prions are, unlike viruses, they're not common, so it's not likely to get a prion disease, I mean, thankfully. And there's some debate about where exactly they came from, but it's generally thought that these are, they originate from defective gene mutations, and these are therefore inherited. So you can actually be born with an unfortunate condition in which these prions will arise somewhat spontaneously due to bad genetics. But then it's now thought that those proteins, if they get out of the original person, can then spread. In other words, you can sort of be born with bad proteins that, by the way, don't really start misfolding until you get older. So they don't stop you from living, but they'll manifest as you get older in life. So, there's a lot of complexity to the disease, but people can be born with that disease that kind of kicks in later in life. And then it's also now believed that if those individuals exist, it's possible for them to spread those proteins from person to person, and then a person who's not born with that could also end up with that disease as well. Now, these are today coming from both infected individuals and also environmental sources. So it's possible that animals can have these and spread them to people. It's also possible that an infected person can spread them to another person. So meat that's not properly controlled could theoretically have them, although I think that's rare. It's possible. Another possibility is during a transplant or some kind of neurological surgery or organ transplant type surgery. So if a person is genetically inherited this disease, if that person is operated on while the proteins are misfolding, it's possible that fluid could be transferred from one person to another and spread the disease that way. But for the most part, it's difficult to transfer that infected fluid outside of certain types of procedures, like a organ transplant procedure or open brain surgeries and things like that. It's possible to transmit them that way. But luckily, these aren't very common. But some of the examples, as a human being, it's called Creutzfeldt-Jakob disease. So as a human being, if you were to acquire either genetically or through an unlucky exposure, you'd have what's called Creutzfeldt-Jakob disease. So that's the way it's described in humans. There are a variety of animal-borne variations of it, but one that you may be familiar with is called mad cow disease. Mad cow disease had a big scare in the early 2000s. A number of people died from it, and there was a lot of regulatory procedures that changed around animal meat, cow meat in particular, And this is one of the reasons why you can't necessarily just go to the store and buy cow brains and cow eyes and cow spinal cord. Not that you probably want to, but if you wanted to, it would be difficult for you to get that because that's the part of the animal that transmits the disease. And so one of the ways that they've dealt with trying to stop that from spreading is just by not allowing conventional sale of the central nervous system of a cow. And so that's one of the reasons why it's hard to get that. And so regular meat, not part of the central nervous system, even if the cow were infected, theoretically wouldn't have those prions in it. So you'd have to eat meat from an infected cow, and that meat would have to come specifically from the central nervous system. So one of the ways we keep that from happening is just that type of meat is not available. But ultimately, that's how people did get mad cow disease, and that's where that came from. A more common example these days that people are monitoring is called chronic wasting disease. And this is one that we're seeing in deer, where elk, moose, deer, reindeer even, all of these can carry a prion that as of right now is being spread just from one type of deer to another. But there's some concern that's being monitored that it's possible that, kind of like mad cow disease, it's possible that one day this prion could jump from deer to humans. But as of right now, that's not happening. Mad cow can jump from cows to humans. Creutzfeldt can go from human to human. As of right now, CWD, which is what we see in deer, it only spreads from one deer to another. It doesn't necessarily spread from deer to humans. But one of the concerns is that the prions can mutate and change over time, and that can start happening. So that's called chronic wasting disease. And you do hear about that in the news every once in a while with the public health agencies monitoring that. So that's the end of part one there. In part two, we'll look at the way that viruses replicate and then ultimately how they cause damage and disease. Going back to the idea that they're like a computer virus, kind of what happens when the virus gets into the cell and what are the various outcomes that you can see there.